Okay, so here's an awkward review that I'm putting together. All right, so stats. um, Start off. One of the first things that you'll need to know is like, what is the null hypothesis? And that should be easy. It should be like a, you know, 230 kind of thing. But the null hypothesis is essentially nothing is happening, right? So the experimental hypothesis is there are two things that are different. The null hypothesis says they're the same. That's important. Well, I'll tell you why it's important in a second. A p-value of 0.03 is essentially going to mean that there's a 0.03% chance that, well, sorry, that's not actually true. There's a 3% chance that your distribution that you think is different from the second distribution is actually from the same one. So there's a very low chance, 3% chance, that distribution is from the same distribution. So there's a 3% chance that the null hypothesis is true. That's what a p-value of 0.03 means, and that's how it relates to the null hypothesis. All right, and then a moderating variable and a correlation. That's pretty, simple, right? It moderates the correlation. So if you're thinking about, what was the example from class? It was game attendance for baseball games when the team is doing well. And so there's no correlation. It's totally flat line. Until you add your moderating variable of, I don't remember what they called it, residential mobility, I think, but essentially it's like how much of someone's life is lived in a city, like the city that they're going to see the team in, right? So I don't remember the city that they talked about, but it was one where everybody's kind of, stuck there and they're unhappy. Like, in Cleveland, everybody loves the Cleveland Browns, even though they real suck. And they're not leaving. There's low residential mobility. And so as the Browns do worse, game attendance goes up because they're going to support the team. I know they're not baseball, but whatever. And then the baseball example where there's high residential mobility is here in Phoenix. People come, they leave. And so they don't have like an affinity for the Diamondbacks. And so then you see that game attendance decreases as the team does worse. So they want to go when the team's doing better. They want to see a good game. They don't really care about supporting the team. Somewhere where people are more invested in their team, they want to go to support the team. So then they go more when the team is doing worse. And then you have two correlations that are essentially like this. The moderating variable shows you that there's two different correlations. If you get rid of the moderating variable, now you've got like a flat correlation. It doesn't always have to be flat and then into, two different types of correlation. It could be that you have like a slightly, a small correlation that turns into a very steep correlation and no correlation. It doesn't matter what the shape of the distribution is or the shape of the correlation is. The moderating variable just means that it changes the way that two variables are correlated, because it's essentially like the third variable problem, but you've identified what the third variable is. Now it's a moderating variable. All right, more stats. Race car. And the reason I say, well, I'm just going to let you try to guess why I said race car. So what does the graph of an experiment with one IV look like? How many bars is it going to have? So I'm going to let you well, I'll give you the answer to this one too. But the graph of an experiment with one IV is going to have one line that connects 2 or maybe 3 bars. It shows you, how they connect to each other. How many bars if you do a bar graph? Well, 2 or more. So you could just have 2 groups and you have 2 bars. You could have 10 groups, 10 bars. It doesn't matter. It doesn't matter how many levels of your IV there is if there's still just one IV. So if your independent variable is age and you say young and old, there's 2 levels of that. Or if you break it down by 10-year increments from 0 to 100, you've got 10 levels of your independent variable. Your independent variable is still age. So it's the same thing, right? If you have 2 IVs, it's going to change the way that the bars hang out with each other, essentially, right? So if you're going to have 2 groups of 2 bars if you have 2 IVs, or you're going to have 2 lines if you do a line graph. So we're thinking about age again and driving. I guess we'll just use driving. We know that we have bars for young people and their ability to stop when they're texting or not. And then we have trying to figure out how my fingers need to go to show you this correctly, but this is like that backwards drawing task with that, HM guy. Anyway, so you have a pair of bars for young people, cell phone, no cell phone. You've got a pair of bars for old people, cell phone, no cell phone. So the way that the graphs are different is you've paired up your bars. You can still have, 5 pairs of bars, 10 pairs of bars. It doesn't really matter. You're going to have pairs of bars now. If you do lines, you're going to have 2 lines. And those can either connect 2 points, they can connect multiple points, but you're going to have 2 lines. That's the difference between one IV and 2 IVs. All right, so these next 2 questions here, the difference between levels of one IV is or N. What you're looking for is either a main effect or an interaction. And the next one, the answer is also either a main effect or an interaction. The difference between levels of one IV that is not identical for levels of the second IV is which one of those? I'm not going to give you the answer to that one. Look it up on your own if you need to. So a lot of you probably already know what it is. That's fine. Well, it's great, actually. So a difference between levels of one IV is the difference between levels of one IV that is not identical for levels of the second IV is what? Okay, next. Main effects and interactions. Which of these graphs to the right is an interaction? And I'm trying to decide if I'm going to give you the answer to this one or not. I guess I will. I don't know if you can see my cursor. Yeah, it looks like you can. It's this one. This is the interaction. It's a spreading interaction. Oh. These 2 are not interactions. But what are they? So this one is showing 2 main effects. You have one main effect because these bars are not in the same place. The orange bar or yellow or goldenrod, or I don't really know what I chose here, some sort of color, but the yellowish one is not in the same place as the red one. It's a little bit higher. So there's a main effect there. The bars are not together. The second main effect is that the ends of the bars are not on the same plane, right? One end is high, the other end is low. And so that's the second main effect. The graph in the middle, this one here, this is showing just one main effect. The bars are not on top of each other. So going downwards, you've got two main effects, no interaction. You've got one main effect, no interaction. And then you've got an interaction. And there's likely, let's see, if you were going to decompose it, there's probably a main effect, maybe. This is one of those ones where you'd have to look at your p-value. I won't ask you about, but you should know that this is an interaction. You should know that there are other types of interactions too. Crossover interaction, there's others. Review the different types of interactions that there are and know what they'll look like in line form, but also know what they'll look like in bar form. Remember the trick to convert line graphs to bar graphs. Essentially what you do is, let's do it on this one here. So you'll draw a bar that goes to just this point right here. So it'd come up, here's one bar. The next bar would go to this point, so it comes up, and here's your second bar. So you've got two bars over on the left. You do the same trick on the right. You got this little short bar here, and you got this medium-sized bar here. So you can derive a bar graph from a line graph. You can derive a line graph from a bar graph. You can plot out either one from a set of numerical data. So review how to do that. What does a 2x3 design mean? There's a lot of information just encapsulated in a 2x3 design. So first, how many IVs are there? Well, there's two IVs, right? There's, well, and then the second question is how many levels of each IV? Well, there's two levels of the first independent variable, and there's three levels of the second independent variable. So there's a lot of information encapsulated in there. The number of numbers is how many independent variables there are. The actual number is how many levels of each independent variable there is. How many interactions could there be? This is also encapsulated in there, sort of, but you kind of have to know how to figure it out. In this case, there's one possible interaction. There's two numbers. There's really only one way that they could interact because you have a number and a number. They can only interact one way. There's one possible interaction. How many main effects? Well, the number of possible main effects is equal to your independent variables. So 2. If you had a 3x3x3, 3 possible main effects, there would be 3x3 by 3. There's 3 possible two-way interactions and 1 three-way interaction. All the different ways that the two numbers can be hooked together, there's a two-way interaction. All the different ways that 3 numbers could be hooked together, there's that many three-way interactions. And then there's a main effect for each independent variable. Next. All right, factorial design. What is full factorial design? Well, it just means that you have participants in every possible condition. So then that kind of ties into the next question is what is each possible combination of independent variable levels called? So if you have a 2x2 design, you've got an independent variable with levels A and B, you've got another one with levels 1 and 2. And so full factorial is going to say that you have participants in 1A, 1B, 2A, and 2B. There's participants in the whole thing. That means that there's participants in each possible combination, which means that there's participants in each cell. So a cell is each possible combination, right? 1A is a cell, 2B is a cell. Then I already said this, but know how to represent data in table, line graph, and bar graph formats. So what that's saying is know how to look at a table of data that says like 5, 10, and then know how to graph a line that goes from 5 to 10. If you have then another set of data for a 2x2 design, know how to graph that, and know how to do it also in bar graph form. So just review the notes from that section. If you have questions, come to office hours. We'll check the email for the updated office hours for this week, or email me. That kind of seemed like that was the end of the slideshow, but it's not. So haha. Okay, so there's more about factorial designs. A between groups design just means that the whole thing is treated like a between groups experiment, like go figure, right? So in a 2x2 design, you've got 4 different cells. You're going to have 120 minimum participants. The exact opposite of that is a within groups design, within groups factorial design. You just treat both independent variables like they're within groups design. So you have the same 30 people in every condition. The minimum number that you need in 2x2 within groups design is 30. And then a mixed design is exactly what you think it is. It's a little bit of both. So one independent variable is treated like a between groups thing. It's treated as though it's a between groups design. And the other independent variable is treated like it's a within groups design. So in a 2x2 design, you're going to need 60 because 30 people are going to go through one entire independent variable, and then another different 30 are going to go through the second independent variable, the second level of the other independent variable. But the other variable, they've gone through both instances of that, if that makes sense. So the mixed design is one is treated between, one is treated within. Let's see if I can come up with some examples. Oh, yeah, okay. So our alcohol photo, planter alcohol photo experiment, and we said, what if you had people that were drunk or not drunk? Well, that would be a mixed design because you would have people go through that were drunk for everything, and they saw both alcohol and plant photos, and you have people that were sober and they saw both alcohol and plant photos. So they're going to see alcohol and plant photos, so they're going to see both levels of that independent variable, but the sober people cannot also be the drunk people. So you have to have 2 different groups. So in that case, your minimum number is 60. How do you graph a 3-factor design? It's a good question. I'm going to let you look it up. You know it's important now, but only sort of important. Take a look at it. See if you can do it. One question about it. So just review that. What makes an experiment a quasi-experiment? Well, it's the use of a quasi-experimental variable. But a quasi-experimental variable is going to be anything that can't be randomly assigned, right? So gender, age, other things, too. Musical preference would be a quasi-experimental variable. A repeated measures design can add a little bit of clarity to a quasi. Well, let me backtrack here. So a quasi-experiment is kind of not great because we can't get rid of that variable that makes it quasi. And that could be like a third variable that pushes an effect that we're seeing. So if there's a difference between men and women, well, it's not because it's hard, it's impossible to assign somebody to be either a man or a woman, right? So there could be other factors, outside factors that aren't just being a man or a woman. It's like the societal constraints around being a man or a woman that push the difference between men and women, not the actual gender difference. So you've got all these potential third variable problems that are inseparable from that quasi-experimental variable. But how can repeated measures design add some clarity? Well, you can measure people at different levels. So you can, if you're looking at something that might change across time, or you think it might change across time, think about our example of sentencing decisions after a meal break. So there could be a difference in the type of case that comes up throughout time. It could be that violent criminals are brought in right before food breaks, maybe. But the repeated measures design makes that less likely. So we have it over the course of an entire day. There's measures that go on. So it is possible that a very violent offender would come in right before lunch. But we're repeating our measures. So it's less likely that you had a violent offender right before lunch and right before tea time or whatever they took. And then it's very unlikely that there was also a violent offender that came in at that last break that they had, right? So it's giving you a better look at the data. It's trying to work out the kinks of the quasi-experimental design. One thing that you need to know about one thing you need to know about repeated measures is essentially that you are measuring your outcome variable multiple times. So you're not just getting one read on it, you're getting several reads on it at different times. So you can be sure that your original measurements are sound because they're gelling with your second measurement, your third measurement, your fourth measurement. That's a repeated measures design. We talked about the inherent problems with quasi-experimental designs, but why do we use them, even if there are these problems? Well, a lot of times it's because you can't do anything else. If you think that there's differences between men and women, yeah, it might be because of the societal constraints that surround being a man or a woman and not the actual biology, but it doesn't matter. That's what you have. You have men and women. You can't really assign somebody, to be in a different group. If you have age, you can't assign somebody who is 60 to be 10. It's real world data a lot of times. So if we had the opportunity to randomly assign people to different conditions, we would. But a lot of times quasi-experimental designs are used because that's all we can do. It's the best we can do, right? The healthcare example, that was real world data that existed. They have to use it. It would be better if we could randomly assign things, but we can't. So doing science on quasi-experimental data is better than not doing science at all. Okay, next, small n designs. The focus of small n designs is different from larger studies because larger studies are looking for, well, we're pulling numerical data, right? We want to quantify a behavior. And then we also want to find the average, the midpoint of that behavior and say, is that behavior different for this group than this group? So we're looking at, average differences. When you have a small n, you might not really have an average. You might have one person. And is one data point really telling you that they're different from a group of like normal people? Not really. So sometimes the focus then becomes the person and the behavior itself, and not the numerical representation of that behavior. So you're going to have things like case studies. You're going to have very detailed descriptions of behaviors instead of a focus on numbers and statistics. Well, and that already answers the second question there, right? You're going to use systematic observation, um, case studies, stuff like that. Sorry, I'm tired. A major problem with small n designs is it's kind of akin to the problems that we have with what we just talked about, quasi-experimental designs, and that's you don't know what third variable problems there could be. If your n is so small that it's 1, and there's some interesting behavior going on, so say Tn, who has blind sight, he can see even though he can't consciously see. Well, we can say that it's caused by meningitis attacking a certain part of his brain or his optic chiasm or whatever, but we're not totally sure because meningitis attacks multiple areas. So logically, it makes sense that it would be the V1, but maybe it wasn't the V1. Maybe it was something in the optical nerve. Maybe it's something in a node of the brain that we don't know is important for processing, cognitively processing vision. So we don't really know the extent of what's going on. If we had a larger N, this kind of messy stuff, if we had a lot of people that had meningitis, the different areas that were affected by that meningitis would become noise. Unsystematic, unintended noise, and that's always fine. Then you could look at where the actual overlap is. You'd be able to see that, everybody who has meningitis damage in, let's say, the V1 has blind sight. So then we can start to pinpoint that. But with only one person or two people, there's so much damage that you don't know what's noise and what's signal. So that's a problem with small n designs. But there are testing methods to help overcome that. In a way. So it doesn't necessarily get to the problem of Tn. Sorry. It doesn't necessarily get to the problem of where is Tn's damage important. We know where his damage is, but where does it become important for blind sight? But for other things, these measures do help. So, and those are things like the stable baseline. The stable baseline is essentially a repeated measures design for the baseline and the later behavior. Repeated measures design is usually just looking at the behavior and not the baseline behavior because you're going to compare two groups. So a stable baseline is taking the idea of repeated measures and applying it to both your treatment and your baseline measure. And then you're going to compare your baseline measure to your treatment measure as though they were two different groups. A multiple baseline design, you can only do this when you have more than one participant, but essentially your onset of when the treatment occurs is different for each person. You can rule out things like a history threat by using multiple baseline design. It's important to note that also encompasses different things you see in the stable baseline design. So a multiple baseline design is a stable baseline design, but you're shifting around when you start treatment and stop taking baseline measures. And then lastly, a reversal design is when you go from treatment to baseline to treatment to baseline to treatment to baseline. You just keep flipping back and forth. So you can figure out if something is working. So a good example for this would be maybe ADHD medication or pain medication. So you could assume that if you want to see that it's working, you could have your patient be on the medication for a few days and have them rate their own pain, or if it's ADHD medication, you can rate how often they go off task, then remove the medicine for a while, see where the behavior goes. Give the pills again, see how the behavior changes, remove the pills, see if it changes back. So you've got this reversal design. There's one thing that I want to mention about reversal design that I forgot to mention in our original lecture, and that's that the reversal periods are different. So it's not going to be 3 days on, 3 days off, 3 on, 3 off, 3 on, 3 off. The reason for that is humans and animals are really good at figuring out 3 days on, 3 days off. Even if you're not cognitively aware of it, you might know that, again, with the pain pill example, in 3 days, and again, you're not necessarily cognitively aware of this, but you know that in 3 days the pain might start to come back. And so then you start to get a placebo effect coming in, and it gets very messy. But if the patient doesn't know, or the participant doesn't know when the medicine is going to be administered and when it's going to be taken away, there's no way to try to predict when it's going to be there or not, because the prediction will be wrong. That's a reversal design. Other analysis techniques, these are just that day that we talked about different kinds of things. When might the average not be meaningful for a data set? So the average is meaningful when you have something that's normally distributed. But if you have something that's not normally distributed, like heart rate or earthquake data, like severity of earthquakes and location of earthquakes, those aren't normally distributed. They don't follow that bell curve. They follow something else, maybe a power law. And that means that the average isn't meaningful then. So when is the average not meaningful? Well, when something is not normally distributed. If you want to analyze something that's not normally distributed, like stock market data or heart rate during a stress test or earthquake severity, then instead of comparing average to like the average of one group of volcanoes to another group of volcanoes, that doesn't make a lot of sense. What you might want to do is a time series analysis. So you can quantify the distribution. You can figure out, is it a power law distribution? Is it a log normal distribution? There's other things you can do beyond that, but it's way beyond the scope of that lecture. So really, I just wanted you to know that sometimes the average isn't meaningful, and there are still things we can do with that. So you don't have to be a slave to assuming that the average is meaningful. And even once you know that the average is not meaningful, that doesn't mean you're stuck. There's other things you can do. You can do time series analysis and other things. All right, what can MDS do? MDS is multidimensional scaling. It's like my thing, right? So it's a dimension reduction algorithm. It can make cool pictures of your data. It can help you visualize your data. But it's also a way to observe the perceptual space, like humans' perceptual space. So that's how Shepard derived the color wheel that he published. There was a color wheel before, probably by some Renaissance painter or somebody even before that, but Shepard's color wheel was confirmed by multidimensional scaling. So it's looking at the perception. How have I used MDS? Well, there's a couple different things, and they're in some of the slideshows that you have access to. Then there's like one or two questions that might sort of touch on this. So if you have time, look it up. If you don't, this might be the thing to skip. But I've used multidimensional scaling to look at the similarity of sounds, right? And so we know that consonants and dissonance are a primary dimension of how sounds are organized. Another thing I've used multidimensional scaling to do is, looking at vowels, right? So we had vowels that are matched for fundamental frequency and intensity. People listened to them, they rated the similarity of the vowels, and then later they rated the pitch of the vowel, even though it was controlled for fundamental frequency. And even though it's controlled for fundamental frequency, people heard a difference in pitch, and that we showed that was one of their main organizational techniques was the perception of pitch for those vowels. So those are two things I've done with MDS that might come in handy. Replication is important because if your study can't be replicated, you may have you might have made a type 1 error. Maybe you lied about your original data. Replication is important to make sure that what we're reporting is actually happening. And then what types of replications can be done? You can do a direct replication, which is just doing the exact same study again, carbon copy. You can do a conceptual replication, which is where you go deeper into the actual experiment and figure out what is the meaning that they're trying to convey. So what is the underlying message? You then apply that message to something else. So if you want to say that comedians from the Midwest aren't funny, then you might decide that, well, yeah, we could do a direct replication. We could look at how often people laugh when listening to a comedian from the Midwest versus the East Coast or the West Coast. But maybe that study's already been done. Now maybe what you want to do is move away from comedians in the Midwest, because maybe you think that it's actually just people in the Midwest don't have good senses of humor. And you want to look at people like, say, on Thanksgiving. How often is a family from the Midwest laughing at somebody's joke versus somebody from the East Coast and the West Coast? That's a conceptual replication. And then a replication plus extension is where you essentially do a direct replication and add an entirely new condition or several new conditions. But you want to replicate and show that it replicates and then extend it into a new area, right? So thinking about the example from the PowerPoint is the note-taking example. People are able to remember new points better when they've written the information rather than typed it. And so you could replicate that and add something else in. What about a typewriter? Or you could replicate their findings of typed and written and add in, like, what if you texted your notes to yourself? And then see if that changes anything. That's a replication plus extension. The replication crisis is just that there's a lot of stuff that doesn't replicate. Shoot, right? So, and that's not just psychology. That's in a lot of areas. Physics is not outside the realm. Mathematics isn't safe. None of us are. It does seem like maybe there's a little bit more in psychology, but not much. The problem is everywhere. And I'm just comparing psychology to sorry. It is late. I'm just comparing psychology to physics and math, other hard sciences. But if you consider maybe political science, social science, and stuff like that, well, there's probably quite a few studies that don't replicate there too. Maybe more than psychology, or maybe less. I actually don't know, but you'd expect there to be about equal. Well, some studies don't replicate for a few reasons. Number one, the replication could be bad. So one of the things that was happening recently was there's a lab that was trying to do a lot of replications, but they were using participants who just wouldn't be good at responding to the questions. So some of them are very specific to, let's say, like African Americans, like, America, in just the United States, there's a different subculture for African Americans than, say, the 2 or 3 Black people who live in the Netherlands. Like I get the feeling that's not a very heterogeneous population in the Netherlands. Um, but that is where a lot of the work was being done. A lot of the replication work was being done is the Netherlands. So what they do is they try to replicate these findings that work with African Americans with these like 8 feet tall Dutch people who all live in, central Netherlands, and it doesn't replicate. Well, maybe it's because you didn't use the right population. So some of it is that. But then sometimes there's problems with the original studies too. Studies from 30s, 40s, sometimes the 50s and the 60s didn't use enough participants. It was harder to get people back then. We didn't have SONA in place. We didn't have the internet for people to get data that way. And so sometimes papers could be published with 10 subjects. That's still a little light for some, but not all. In fact, if you read Cherry's 1953 article, I think it is, on the cocktail party effect, it actually might be from the 40s, but he just does a couple of experiments on himself and says like, oh, and here's an interesting thing. That's it. The N is 1, and it's not even like some of those small N studies that we read about. That would be a problem with the original study. Not enough participants. Sometimes people outright lie, but then sometimes they don't. Sometimes they hypothesize after their results are already known, which means they see that something becomes significant, and then in their write-up they say that that's what they predicted all along, was this thing that ended up becoming significant. And then there's also p-hacking, which we've talked about a few times, but that's adding more people to your subject pool until your p-value dips below 0.05. So it's at 0.08. Let's add more. It's at 0.07. Let's add more. Oh, now it's at 0.1. Let's add more. Now it's at 0.05. Good, publish it. That's p-hacking. So sometimes there are problems with replications. Sometimes there are problems with the original studies. But once the problems with the replications were fixed, it's still about 20% of hallmark psychology studies that don't replicate. So it still is a crisis, even though it's not as bad as it looked originally. How can we get past those problems? Well, we can make sure that we have good sample size. You can report all of your analyses and variables. So instead of this prevents harking, hypothesizing after the results are known, you show everything you looked at and you show how you looked at it. Because sometimes you can do an ANOVA and then one thing doesn't really work out. So if we got rid of it and did a t-test, that would be better. But if you report all of your analyses and variables, you'd have to show that you did an ANOVA and a t-test and a two-way ANOVA and whatever. Published data, not really sure what I was talking about there. Oh, no, I know what I'm talking about. You publish the raw data, so not just the aggregate data. Usually, right, in papers, we publish the average, the standard deviation, and the statistical test. But another way to overcome some of these replication problems is just to publish the actual raw data. So this would dissuade people who were making things up. It would dissuade people who knew that their data were kind of bad and chose a certain kind of analysis just to show what was going on. It allows for other people to look at your data later and maybe make a new conclusion that you didn't even know was there, too. And then the last thing that we can do to overcome the problems of the replication crisis is pre-register. And remember, we talked about that not too long ago, that all that means is you tell the journal what you want to do and how you want to do it. They give you the okay, you do it, and then they're going to publish it whether it is significant or not. So you can't do anything fishy because you've already told them how you're going to do it before you collect the data. And then we got 2 slides left. Are they both about meta-analysis? No, they're not. So meta-analysis, what is it? Well, essentially, it's you grab all of the studies or a lot of them on a certain topic, and you want to know what is the actual effect size of this topic. So you're gonna, combine all of the different t-values and f statistics and stuff like that, and correlations, and figure out over all of these 10, 20, 100 studies, what is the true effect of what I'm interested in. So one of these was lying, and it takes time. And those studies show that in a whole bunch of different conditions, when participants were instructed to lie, it took them significantly longer to respond than when they were told to tell the truth. In some instances, there was a large difference. In some instances, there was a smaller difference. But when you do the meta-analysis, essentially it averages all those and says that there's a moderate difference in reaction time for lying versus not. Moderate but reliable. So now we know that lying does take a little time. The file drawer problem is that a lot of studies that show null results just don't ever get written up, or they get partially written up and then they go in a file drawer. They sit on somebody's computer. A good example of this is when we're looking at those depression studies, like do depression drugs actually work? Well, published studies say they work about 80% of the time. But if you are able to get all the data from all the studies that have been done, including things that were never written up, depression medication works about 50% of the time. So that's the file drawer problem in action, is that we would know that depression medication is kind of like flipping a coin, when right now we think that, 80% success, do it. So we'd know more of the truth if there wasn't the file drawer problem. And then the last slide is just a couple things I couldn't figure out where to fit in. First, what is ecological validity? Well, it's making sure that your stimuli are as close to the real world as possible. So if you can have somebody be in the real world and do tests on them, that's great. But if you can't have somebody in the real world, what can you do to make your lab or whatever testing environment you have seem real? How can you make your audio stimuli sound real? How can you make your visual scene look real? These are all questions for ecological validity. The idea behind it is that we live in the real world. And so if you bring people into the lab, they might not be able to function as well in a lab because it's not the real world. There's things that aren't there that are usually there. So if you can test people in the real world or very real conditions, you might see different behaviors. And then last, why are psychology participants weird? Well, because they're Western, educated, industrialized, rich, and democratic. We all come from essentially, the same background. One of the things, you can assume that that's true for Americans, it's true for Germans, it's true for people from the UK, and this is where a lot of our major universities are. The US, Canada, the UK, Germany, all of Western Europe, Australia, places like that. We're all basically the same. Yeah, sure, Australians have funny accents, but aside from that. And what's up with the South African accent? That's just totally crazy. Like, I don't know. It sounds weird to me. Like when you're watching District 9, you're like, what are they saying? So anyway, even when you consider that there is a lot of scientific work coming from Japan, well, they're just not Western. but they are educated, industrialized, rich, and democratic. So they're not ticking one of those boxes, but there's a lot of similarities between cultures. So you can play that game by looking at different countries where research is coming out from. There's a lot of homogeneity in the type of people that are doing the work and the type of participants we get. Not a lot of work comes out of sub-Saharan Africa, right? Because there are other problems that are going on there that we just can't. There's not the free time or the resources to do, psychological research like we do in weird countries. So that's what it means. It's like psychology participants are weird. It means that if you're ever going to generalize, it's really just going to be to weird participants. If you want to know the true state of the world, you can't unless you go out in the field because you have weird participants. Okay, that is pretty much everything I have for the review. But like I said, I'm just not there tomorrow or today, Tuesday, for our class. I'll be back around 4, maybe 3:30. Come find me in my office and ask me questions if you have them. Come find me after my first class. It gets out at 11:45, so I'll be in my office from 11:45 to 1, not 1:30. So you can ask me questions then. You can ask me questions by email. You can also ask me questions. I'll be in Thursday morning in my office. So from about 8:30 to 10:30, I'll be there. From 11:45 to 1:30, I'll be in my office. And then we have the test, so it doesn't really matter after that. But there's all those different places to meet me if you have questions. Well, I mean, it's all the same place. It's all in my office, but there's different times. Or you can email me, and I'll answer your questions. So hopefully this was helpful. Answer for yourself some of those questions I didn't answer for you. And then come to me with your questions, and I'll see you on Thursday for an exam.